All right, thank you, Arlene. And I'm very excited today to be kicking off this DILI-sim Applications webinar. Really, we're here to emphasize and focus on a dialogue and a discussion around the practical applications of the software, and to hear from some excellent panelists. So thank you all for joining us today. We're just elated to have you. I'm Brett Howell, president of the DILI-sim Services division of Simulations Plus. As Arlene mentioned, we have uh, Dr. Vlad Coric, Dr. Paul Watkins, and Dr. Lorenzo Pellegrini, who will be joining us today for a panel discussion. So in just a few moments, after I go through a few brief slides, I will ask them to introduce themselves and just to say a few words about their participation in the webinar, and then we'll actually go through a series of questions and have more of a discussion and a dialogue. So you'll hear from them briefly. Before we jump into that, just a couple of notes on Simulations Plus, who we are. We are a global modeling and simulation company focused on quantitative solutions for the pharmaceutical space, but also the chemical space and other industries as well. We really specialize in a few different areas. Chem informatics, or also known as machine learning, is one area. PBPK modeling, physiologically based pharmacokinetics, is another area of expertise for us. Population-based pharmacometrics, so POP-PK and PKPD modeling, and finally quantitative systems modeling. And today, of course, we're going to be talking about quantitative systems toxicology in the liver safety space. But really, at Simulations Plus, we are an end-to-end, comprehensive solution for your modeling and simulation needs, and we don't just provide services, which is one thing that we do emphasize, and I'll talk about today with a number of examples scenarios, but we also license a lot of software platforms. Many of these you will know, household names like ADMET Predictor and GastroPlus and Monolix, and so these are platforms that we license to the pharmaceutical industry around the world. And we'd love to work with you or your company and collaborate in either of the software or the services area, in any of these different applications of modeling and simulation. We also pride ourselves at Simulations Plus on having solutions that work and partner well with development companies from discovery all the way out through post-approval. So some of our discovery-focused applications like ADMET Predictor and MedChem Designer are sort of best suited for that early phase. Some of our products are best suited for sort of the middle phase here, and then more some of the pharmacometrics tools more for the latter phases, in the clinical phases and beyond. But really, what you see is a seamless bridge between all of these and the entire drug development spectrum, and our services and our regulatory interactions as well, and services in that area really span this entire spectrum. So we've put together, between our 4 different divisions around the globe, which are based, by the way, in California, New York, North Carolina, and then also in Paris, France. So we put together a nice combination of tools and services. Now, our division, DILIsym Services, is, as I said before, a mechanistic modeling focused group, and we really got our start in the liver safety space, and that's going to be where we focus our discussions today. But we're doing a number of other things now, and just to put those things on the radar, we are focusing now on kidney safety as well. We are focusing on models that really look at efficacy and pharmacology, things like idiopathic pulmonary fibrosis, NAFLD and NASH, heart failure, kidney failure, and also radiation syndrome. So we've really focused and moved into the efficacy side of the business as well as the safety side. But again, today our real emphasis is on liver and the practical side of DILIsym, how it's used, and examples of how others have applied it. Now, if you're new to DILIsym, if you haven't heard of it before, or you haven't had an introduction to us, you may be saying, what would you even use this kind of software for? Well, before I give you some practical examples, I just want to lay out some high-level concepts around this. So you may want to predict DILI liabilities before they occur. So this is the sort of the gold standard for modeling and simulation, the prospective prediction, and we certainly are using DILIsym for that in a number of cases. There's also this relative comparisons that you can make when you're choosing a lead candidate for development. So how does candidate A versus B versus C look on a relative scale in terms of liver safety? And then lastly, we've done a lot of investigative toxicology, I would say. So examples with sponsors where there's some issue either with their program or a related program, and they're interested in following up, understanding mechanisms, understanding the possibility of mitigation through alternative doses, and all these types of investigative toxicology questions that come up. And modeling and simulation is great for that as well. Now, this whole platform, DILIsym, got its start with the DILI-sim Initiative, which Dr. Watkins really headed up and started about 11 years ago now. And the DILI-sim Initiative has been supported by pharmaceutical companies, over 20 now, mostly larger companies, over the last 10 or 11 years, and they've provided resources for the development of the software. But really, it's served as a public plus private partnership, because the academic groups and the FDA has been involved all along the way. So actually this consortium started with the CRADA with the FDA. We've had various FDA members involved in the board, and scientists involved in helping build the model as part of the team through ORICE fellowships and different scenarios. And currently the FDA is a licensee of the software, using it more for determining, and evaluating submissions that come into them, and companies will submit where they are using the software to address a question. So it's really been a great forum for development of this type of platform, especially a platform that's in the safety space, which we typically, see as a less competitive space and more pre-competitive and sort of collaborative. So it's been an excellent forum for that. And I should mention, we've now brought Remdesivir into the fold, so kidney safety is a part of this as well. So just to lay the foundation, what is quantitative systems toxicology? Well, it starts with a basic mathematical framework of a particular disease or application or area. And in this case, you can see here on the screen, DILIsym is a basic model of liver function and liver dysfunction, and that's where the starting place begins. And then we layer on top of that drug exposure in the liver. Of course, we use GastroPlus, the gold standard for PBPK modeling, to predict our exposures, concentrations at the site of action or site of toxicity. And then lastly, we have to assess the potency of compounds on particular mechanisms or particular pathways. And we do that through in vitro assays, sometimes through machine learning models as well. And that allows us to connect the drug's action with its exposure, with the function of that organ or that system. And all those things come together, then you can actually predict clinically relevant and in vivo relevant toxicity, not just an in vitro signal that may or may not matter for a real patient, but in a particular patient population for a particular individual, would they experience a toxic event or not from this drug and the drug's exposure and the duration of exposure. So now, before we move into our panel discussion, which I'm really excited about, I want to go through just a few snapshots of DILIsym examples. And so there's a lot more detail, obviously, behind the scenes in terms of how DILIsym works and how you can use it. And we're happy to provide training and additional resources for all those things. But really, the focus of today is just practically what questions have we helped companies address or sponsors address. And so this first question is focused on a comparison of potentially new molecules that are being developed, in this case CGRP receptor antagonists that are being developed for migraine treatment and have been developed for migraine treatment, versus a prior compound in the similar space that failed due to liver toxicity. And so we did this collaboration in collaboration with Biohaven. We looked at Tokajapan versus 4 other CGRP receptor antagonists. And what you can see based on this high level graphic is we have predicted all 4 of those molecules to be much safer than the Tokajapan molecule that failed in clinical trials. And so far, all of the clinical trial results that have come out for these molecules have agreed with that assessment. So it's really a confidence providing simulation scenario to say, do we have another reason, another data point, another way to look at the possibility that these newer drugs are safer than, these newer compounds are safer than those developed in the past. And that's really a great example of more of a prospective prediction for future events and to help with decision making. And you'll hear more about this as we go into the panel discussion. Another example, which is slightly different than this, the key question from the sponsor was, we're seeing some liver events, some biomarker signals with our compound, which in this case was solithromycin, a macrolide antibiotic. And they asked the question, do we believe that other macrolide antibiotics in the field have had liver events through the same mechanism? In other words, just because they're somewhat structurally similar, do all these macrolides do the same thing in the liver? And the interesting answer to the question was no, they actually all have different mechanisms and different effects. But generally, you can see the utility here of understanding how a drug is interacting with the liver and how the differences in the mechanism may actually lead to, you know, even larger differences in terms of the clinical manifestation, in terms of immune mediated possibilities and all of those things. So this was a very interesting example where we analyzed mechanisms. It was presented at the company's FDA AdCom as well. Next, we did a project in collaboration with Allergan and Merck where we were again looking at CGRP molecules and comparing ubrelvy to Tokajapan, and very similar outcome as the prior first CGRP example I mentioned, where we predicted that Tokajapan would be a liability. Of course, it was clinically, but more importantly that ubrelvy would not be, and you know that was proven in the clinic, and ubrelvy was approved for use, and I should have also mentioned Remdesivir was also approved, you know, from the first example. So two drugs that were predicted to be safer than Tokajapan subsequently approved by the FDA for use. And what's interesting about this example is that we use completely different data sets than the first CGRP example that I cited. So completely different data sets in terms of the in vitro data that we had to source to feed the model, and yet we got very similar qualitatively responses, which is, you know, of course, comforting to see with this type of approach. Next, we looked at a compound that many of us have heard of in the news, not too in the recent future, and that is Remdesivir. Remdesivir in development by Gilead has been pivoted more towards COVID-19, and what we were really focused on in this example was helping to assess there are some liver safety signals for this drug in the clinic. Why are those occurring? And interestingly, we found that we don't believe they're occurring from the mechanisms that we typically look at and analyze in DILI-sim. So it was a negative example, an example of something else out there is probably leading to these. It could be an on-target effect, it could be something else, but it was a mechanistic analysis, and it was certainly submitted to regulators. Next, I'll talk about the two drugs that we analyzed intended for autosomal dominant polycystic kidney disease, and Dr. Pellegrini is on the panel. We'll hear more from him later. But we really compared lixivaptan to Tolvapden, and it was this, again, key question of between two molecules that are structurally similar, you know, there's a lot of clinical experience with Tolvapden. How would lixivaptan compare? Tolvaptan has some known liver safety liabilities and signals. Would lixivaptan be similar or different? That was really the key question. And you know, you can see with this E-dish plot here that we're summarizing the results that we predicted. This actually published that lixivaptan would be less likely to cause liver events than Tolvaptan. So that's again a prospective prediction meant to provide another data point into the question of the differentiation between molecules with respect to safety. This next example, the key question was a little different. It was about the possibility that acetaminophen could actually cause, you know, could be a carcinogen and cause cancer. And so we did an assessment in collaboration with the Consumer Healthcare Products Association, and this was done as part of a much larger study looking at epidemiology, looking at in vitro studies, looking at animal studies to address this question, and then to submit that overall scientific study to the to the California Office of Environmental Health, who was actually assessing and asking for input on this question before they sort of held a meeting to discuss it. And I believe that's still going to be held in the future. So what we actually were able to do is look at the mechanism of acetaminophen's liver effects, which is a well-known liver toxicant at very, very high doses, but relatively safe and causing minimal issues at therapeutic doses. And what we found, if you notice with this population-based simulations where we see glutathione as an example in green at reasonably sufficient levels for a therapeutic dose across a whole population versus, you know, individuals who have depleted levels at, you know, much higher overdose levels up to 70, 80 grams over 5 days. What we found was that acetaminophen sort of has not much, there's not a lot of space in between the therapeutic and safe realm and the overdose case where there's liver injury for a smoldering low-level injury to occur over a long period of time, which is typically what you need for carcinogenicity. So we really don't think that the mechanisms support the idea that acetaminophen is, you know, carcinogenic. and that it would cause the stresses you would need for that at the doses that are actually used in practice clinically. But it was a different sort of project with a different really goal and focus. Next, we did a mechanistic study focused on pexandartnib, and the question there was really, could we help explain what mechanisms may or may not be accounting for liver safety signals? And this was part of the FDA's review of the drug. We also, these last couple that I'll highlight briefly, are a little different. We have focused in the past and continue to focus in some cases on biomarkers. And here, with this GGF2 large molecule, the real emphasis was on helping to explain or estimate the amount of liver loss that occurred in patients that did have ALT elevations. And this was a published study that really suggested that even though the ALT levels were relatively high on an upper limit of normal scale or a maximum scale, based on the kinetics, the actual risk to the patients was low because they had sufficient liver function. They were not close to what we call a Hy's Law case where there's serious liver dysfunction. So it's really understanding biomarker signatures better is a real goal of a project like this. And in fact, in a similar project that we did focused on a compound, a large molecule meant to treat radiation syndrome, we looked at these fairly unique ALT elevations or profiles where these patients had extremely high levels of, not maybe not extremely, but very high levels, over 1,000 units per liter of ALT, but in a very short time period. If you notice the x-axis here, it's just a few days. And what came out of this study in this important publication that we put together was that you really have to look at the details of the kinetics of biomarkers to understand what they're telling you. And many of the traditional metrics, the traditional ways of looking at liver safety biomarkers don't include the kinetics. So you can actually make, conclusions or draw conclusions that aren't giving you the real story. So that was a biomarker example that we did as well. So the take-home message from my quick overview is that DILIsym has helped many drug developers and investors with key decisions and submissions. We can help your program or your project as well. We offer consulting, but also through our consortium membership and our direct licensing program, we offer licensing of the platform. So your expert modelers could take the platform in-house and do some work on it as well. So let's shift over and let's have a good discussion with our panelists. So first of all, I want to thank all of you while you're joining. I'll stop sharing my screen and allow you to jump on camera. We're going to go through some introductions from each of our panelists and some introductory comments. And then once we've gone through that process, I'll be asking a series of questions. So, for all of you, maybe we can start with Dr. Coric, and then we can go to Dr. Pellegrini and Dr. Watkins. To all of you, just please give our audience a brief introduction to yourself, and feel free to make any additional comments on your participation in the webinar in general. And then after that process, we can go to the questions. So, Dr. Coric.
Hey, good morning, everyone. I'm Vlad Coric, and I'm the CEO of Biohaven. I'm a physician by training, and I've been in clinical research and drug development for almost 20 years now. And I want to thank Brett and Paul for inviting me to be part of this panel discussion. I think one of the reasons I was invited is that over the last 5 years at Biohaven, we've incorporated DILIsym into 4 of our lead programs. And it's really been a critical aspect of each of the programs, and in particular on the CGRP antagonist platform. I know we're going to get into some of those details in the additional discussion, and I look forward to that.
All right, Dr. Pellegrini.
Hi, thanks, Brett. Nice meeting you all. Thanks for the opportunity to share my thoughts today. My name is Lorenzo Pellegrini. I'm the founder and chief operating officer of Palladio Biosciences. By way of quick introduction, I have a PhD in biochemistry from the Max Planck Institute in Germany, a postdoctorate in cell biology at Yale University, an MBA from Wharton. And then I spent 12 years at a leading biotech venture capital firm in the US, where I helped start 5 companies. And Palladio is my 6th biotech, and also obviously my favorite. And just very recently, Palladio was acquired by a larger company, Centessa Pharmaceuticals, and continues to operate as a subsidiary of Centessa. And so my participation on the webinar today is due to our use of DILIsym very early on in the story of in the development of Palladio, and it really was instrumental in our success and many of the good things that happened later on to Palladio. So I'm looking forward to share that experience with you.
Dr. Watkins.
Okay, so I'm Paul Watkins. I'm professor at the University of North Carolina, and Brett said I'm a clinically trained hepatologist who's consulted widely for the pharmaceutical industry. I don't believe any major pharmaceutical company in the world has not consulted with me at some time in the area of liver safety. And the DILIsym Initiative really rose out of my Institute for Drug Safety Sciences and is now funded through 2023, continuing to build the software. I don't have any equity in either DILIsym Services or Simulations Plus. But I'm very excited with the progress that DILIsym has really made and now is increasingly becoming part of decision-making in drug development. And really what is happening is it's following the path that was established in drug-drug interaction. So now I think it's universal that companies will see whether their drug or major metabolites inhibits a certain cytochrome P450 or transporter, and together with exposure information, PBPK, make predictions, essentially rule in or rule out drug-drug interactions. And this has really improved the safety of drugs. It's improved the efficiency of drug development in that it obviates the need to do some drug interaction studies. And really, this is exactly what's going on with DILIsym as an example of quantitative systems toxicology. You measure the ability of the drug, in some cases major metabolites, to inhibit certain pathways, adverse outcomes pathways. Together with PBPK, you put it together and you predict the liver safety liability of the molecule. And it's important to note there is no path towards FDA approval for this. So for instance, GastroPlus never went through any official approval. It just starts being used by FDA, the pharmacometrics group, and then just becomes a standard. And that's what's happening now with DILIsym. The FDA has the license to the software. They've really been very receptive to the modeling as part of the aggregate information. And it's important to note that both ubrogepant and rimagepant were approved without even a liver safety warning, when first in class Tolvapden and second in class MK3207 both were terminated because of significant liver safety concerns. To my knowledge, that is a first. So I've said enough. I'll be available for questions.
All right, well, thank you guys. Yeah, I meant to mention as well that none of our panelists were compensated for participating in the webinar today, so I just want to mention that. I think we'll start with questions, and this one maybe we can start with Dr. Pellegrini and then go to Dr. Coric. This was really more for you two. Why did your organization engage with the DILIsym Services division of Simulations Plus to undertake a DILIsym consulting project?
Yes, thanks, Brett. So let me first give you a little bit of background about what the company is doing. So at Palladio, we're developing lixivaptan, a vasopressin V2 receptor antagonist. We're currently in phase 3 for a large orphan disease called autosomal dominant polycystic kidney disease, or ADPKD. ADPKD is a terrible inherited genetic disease. It definitely deserves better therapies. And there is only one FDA-approved drug to treat ADPKD, and that's a drug by Otsuka called Tolvapden. And as the name suggests, Tolvapden is another vasopressin antagonist, just like our drug, lixivaptan. So the very simple idea behind Palladio is that if Tolvapden works for ADPKD, then our lixivaptan will work too. And in terms of differentiation from Tolvapden, the differentiating avenue for lixivaptan was very clear from the beginning. Tolvapden causes idiosyncratic drug-induced liver injury, as you mentioned in your introduction. And as FDA described it, it is an inherent hepatotoxin. So it is only available in the US under a strict REMS program, as you would expect, which presents an obstacle to its wider adoption. And despite all of these limitations, the sales of Tolvapden for ADPKD last year were $620 million in the US alone, and it was just its second full calendar year on the market. So there's a great medical need in ADPKD. There's urgent need for new therapies. All of this is confirmation of that. So Tolvapden's toxicity gave us a great opportunity to develop lixivaptan as a liver-safe alternative to Tolvapden, and that's where DILIsym came in. So very early on, after starting the company, Palladio, we looked for capital-effective ways to demonstrate that lixivaptan is safer than Tolvapden, and we quickly learned by talking to experts that standard traditional preclinical toxicology experiments are completely unable to predict idiosyncratic drug-induced liver injury. But eventually we came across a publication by your group, Brett, showing that DILI-sim had replicated the hepatic toxicity of Tolvapden very accurately. And it was all there for us to see and to study. And this made DILI-sim really stand out for us because it was a validated tool for our specific case. We had considered other approaches, including certain cell-based assays, but none of them had reached the level of validation of DILI-sim. And plus, when we did due diligence on DILI-sim, we learned that FDA was using it, that industry was using it, that academia was using it. So we concluded that we did not have to create an audience for our DILI-sim result. The audience was already there. And just a final comment on the perspective from a small biotech. So you're in chronic fundraising mode. You're always one experiment away from folding the company. So we had to be very selective about which experiment we were going to conduct. You never want to run an experiment where the outcome is asymmetric. In other words, where you get penalized if the results are negative, but you're not rewarded commensurately if the results are in your favor. For example, suppose we decide to test the fundamental hypothesis behind Palladio. So here we are putting the company at risk, right? The hypothesis is that lixivaptan is safe on the liver. And we decide to test that in a non-validated model. If we show that lixivaptan is safe in that non-validated model, then our audience will say, so what? It is not a validated model. You have not proven anything. But if we show even a suggestion of toxicity with lixivaptan in that non-validated model, then the audience will say, aha, so it's not safe after all. So you get stuck with, you cannot win in an asymmetric outcome experiment like this. And this is why, so DILI-sim was the only system, because it had characterized Tolvapden accurately, the only platform where we knew we could win because of that prior history with Tolvapden. So it was almost a no-brainer for us. It was really the only way to go.
Dr. Coric?
Great, thanks, Brett. So, I think when you look back and when we started first working with DILI-sim, it was around our CGRP antagonist program. And I think both you and Paul alluded to some of the challenges in the field, but it is worth kind of getting into some of the details historically about what plagued the CGRP small molecule field in the treatment of migraine and the first generation agents. You mentioned Tokajapan. That was Merck's first generation agent. That had very significant elevations in bilirubin and ALT in their early phase 2 program that ultimately was the demise of that compound. And then what happened after the Tokajapan experience is, people at first wondered, was this on target, was it off target, and tried to understand what was behind the Tokajapan's hepatotoxicity. We and others in the field had already started to engineer out from a chemistry perspective what we thought was the culprit causing the issues with Tokajapan. And then you saw both Merck and then Allergan and ourselves generate second and third generation molecules that everyone believed would be safe. Now, at the same time, you saw a bunch of other companies decide they were going to target CGRP using monoclonal antibodies with the thought that they wouldn't have liver liability. And so there were two parallel efforts going on. So it was increasingly important, I think, to dispel that A, this was on target, and then B, to show that the second and third generations did not have those propensities for hepatotoxicity. And so then you're left, well, what's the best way to gain your confidence around that? And obviously, given Paul's work in the field, we had gained a deep understanding of DILI-sim and saw how it could bring us value in selecting the right assets to advance and develop the program in the right way. And we made a very significant decision, which looking back, I think actually was the most critical and important decision. That was to use DILI-sim to predict whether our second and third generation agents would have a different profile and not have the liability of Tokajapan. What was really valuable from DILI-sim wasn't just showing the difference of these different candidate agents, but also, it first started with understanding why did Tokajapan have the issues that it did, and the team at DILI-sim really helped us understand there are a number of variables, including that there was this high accumulation of Tokajapan in the liver. There is a very high affinity to BSEP, and what is probably one of the most important factors, Tokajapan is a mixed inhibitor of BSEP and has a non-competitive component where bile salts accumulate and the drug is not displaced, further exacerbating the hepatotoxicity. So that deep understanding of why Tokajapan got into the issues that it did then allowed us to not only show the second, third generation compounds were different, but also explain why. And that becomes really important, I think, to gain patient confidence, physician confidence, and regulatory agency confidence that you have a drug that's not going to have the same type of liability. And the DILI-sim model really delivered incredibly in showing us that none of the second or third generation molecules, either ours or Merck/Allergan/now AbbVie's molecules, were not predicted to result in bilirubin and ALT elevations. And so there was a prediction that there'd be no Hy's Law cases with the second and third generation molecules. And that gave us the confidence to say we had chosen the right molecules, but then also to aggressively develop those and to come up with the right clinical development plan now that we had confidence around safety. And we knew that DILI-sim was the first step, and then you also had to show the clinical results, and that would also validate the DILI-sim predictions. And so we decided to run some very large studies with a large number of patients in order to show unequivocally that you would not run into the same issues that Tokajapan had run into. And looking back, I think Paul alluded to, both our drug and our competitor's drug were approved without any liability on the liver, and that was clear from both very large clinical data that validated DILI-sim. So I can't emphasize enough the value and the importance of incorporating DILI-sim early in one's drug development programs.
Yeah, thank you guys. Excellent, excellent insight there. So a follow-up question, and maybe for this one we could start with you, Dr. Watkins, and then go around, and you guys may be able to provide detailed insight on this, or you may want to be much more general depending on what you're able to share. But I'm sure a lot of the audience is curious, if you have any thoughts or insights on how regulators have reacted to DILI-sim submissions, either from let's say from your programs for Dr. Pellegrini and Dr. Coric, or for Dr. Watkins. In your case, you've seen lots of programs where DILI-sim has been utilized, and just in general, if you have any thoughts to share that the audience can hear on how regulators have responded to these submissions.
Yeah, I can start. So right from the get-go, the FDA was involved. Janet Woodcock was a great supporter of this QST for DILI project from the beginning. And the FDA has supported ORIS fellows who are embedded actually in the team. And the first ORIS fellow actually then went and is a full-time employee in the pharmacometrics group at the FDA. And the pharmacometrics group has the license for the software just recently renewed. And I think they're, I think they are very open-minded to it. And again, understand that it's part of the weight of evidence that would support liver safety. But certainly in my interactions in drugs, such as the drugs we're discussing here, it's been viewed as a helpful and I would say important addition to assessing liver safety. So I would say the reception's been good, very good.
Dr. Coric?
Yeah, I think when you look back to, going into our regulatory filings, we wanted to make sure that we address any concerns proactively around the first generation agents, in this case, Tokajapan. And I think going in with the strength of the DILI-sim data, showing that it predicted these compounds would be safe, and then the clinical data to bolster that, and mechanistically being able to describe why these agents were different than the first generation is really compelling from a perspective of your NDA submission, because really our objectives and the regulatory agencies are the same here. You want to optimize efficacy and minimize side effects and minimize any chance of true hepatotoxicity. And I think it was a very powerful package, starting with DILI-sim, and then, as Paul said, that's one part of the package. Then augmenting it with the full clinical data is a compelling story.
Any comments from you, Dr. Pellegrini?
Yes, I agree with everything that's been said. And I think in our case specifically, we even discussed sharing the parameters of our simulation with FDA so that they are perfectly enabled and capable of running their own simulations on lixivaptan and see for themselves. And I think historically, FDA has always been very, very conservative about idiosyncratic drug-induced liver injury. So it's a kind of approach where it's a kind of situation where you don't want to leave any stones unturned. You just hit them with everything that you can possibly put together, including DILI-sim, but obviously including the clinical results. And I don't have evidence for this, but it's actually an interesting thought. I wonder whether, because you are providing DILI-sim results, whether that will allow you to have a discount on the extent of clinical evidence that you're providing. Instead of running a 10,000 patient study, which would be impossible for ADPKD, for example, can we get away with just 1,000? Because we have, as Vlad mentioned, the scientific foundation and the mechanistic foundation for why our drug will be safer. So it kind of reduces the amount of clinical evidence that you have to provide. Still, you have to provide clinical evidence. FDA, clinical evidence is king with FDA. There's no exceptions, right? But if you can actually shave off just even a little bit on the amount of clinical evidence that you have to provide, it results in massive savings in terms of cost and time. So it's definitely something that you want to have when you approach the FDA. A DILI-sim simulation that provides all the thinking, all the rationale behind why you're safer.
All right, thank you. Well, sort of a related question. I'm sure there are going to be those in drug development listening in to the webinar and saying to themselves, well, I still have some hesitancy. I still have a little bit of fear or a little bit of concern about, approaching not just DILI-sim, but let's say generally new technology, new ways of thinking about this problem from a risk perspective. That's going to be more of a risk to my program than a help. And I don't know if all of you have any thoughts or comments on this. Maybe we can start with Dr. Coric. If you have any thoughts on those who may be feeling that level of hesitancy from your perspective, having gone down that road, Biohaven to some degree, having blazed that trail, what would your thoughts be?
Yeah, no, you're right. And there's always that tendency that people always have the debate, like, should you do this? Should you not do it? What are the risks involved? And I think one of the key factors is obviously you're using a highly validated technology here in DILI-sim. So assuming that that's what you're dealing with, I think the other risk you really have to, put aside. And that, look, in drug development, we need additional, data to make good decisions. And, ultimately, our ability to develop safe drugs is going to rely on having new technology, new tools like this in our repertoire, right? The whole purpose here is to minimize late-stage drug risk, failures, and most importantly, ensure patient safety. So, whenever I hear those naysayer, discussions, I say, look, we don't have a choice here. You have to use tools like this, right? So we can improve the way in which we develop safe drugs for patients, and that has to be our number one priority. So you can't just stick your head in the ground and say, look, we're not going to do this because of risks and we're worried about what it's going to show. Run the experiment, get the data back, be informed by it, and then adapt your program as appropriate.
Yeah, I can comment to that also. I think it's a philosophical issue, right? Do you want to tackle the risky question early on? Do you want to know the answer, or are you going to continue to develop your drug for 3, 5, 10 years, hiding away from the actual issue, right? If there is an issue, you need to know, and that's probably a philosophy that is more predominant in small companies like Palladio, where again, if the fundamental hypothesis is not valid, is not verified, then you just go out and do something else altogether, right? But I think there was another very interesting aspect, a lesson from our experience with DILI-sim that I would like to emphasize. perhaps when we approached the tool, we were ignorant about it, but we were surprised that the results, the DILI-sim results for lixivaptan were much more informative than a simple assessment of toxic versus non-toxic. So one of the most interesting aspects of our result was the qualitative differences between lixivaptan and tolvaptan in terms of which mechanisms of toxicity were most relevant for each drug. And so by doing this exercise, we found that the mechanisms of toxicity with tolvaptan may actually be aggravated in ADPKD patients because the ADPKD mutation may make hepatocytes more susceptible to injury by the two liver toxicity mechanisms of tolvaptan. So this was all very interesting. It provided an explanation for why idiosyncratic DILI is generally not recognized with tolvaptan's use in non-ADPKD patients, non-mutated patients. And another example, for some time at Palladio, we were wondering about the safety of certain metabolites of lixivaptan to the point that we were considering an investment in ways to reduce their levels in people. But our DILIsym simulation of lixivaptan and its metabolites clearly showed that the metabolites of lixivaptan are not likely to give rise to toxicity, unlike the metabolites of tolvaptan. So, we no longer had to do all those activities or make all those investments. The DILIsym results helped us understand, in general, the general framework to develop lixivaptan as a safe drug, including the dosing ranges and including what to watch out for. It informed us about certain potential risk areas with lixivaptan that we might have overlooked otherwise, and we're better off as a result. We can also now develop lixivaptan in a much more aggressive manner, more conclusive, more persuasive manner, tackling the risks early on. For example, we are conducting a clinical study in patients who experienced liver toxicity with tolvaptan and had to discontinue tolvaptan because of the onset of liver toxicity. So we enroll these patients, we give them lixivaptan, and the idea is to show that they can take lixivaptan safely. And this will be obviously very strong, very persuasive evidence that lixivaptan does not cause drug-induced liver injury. And I don't think we would have run this experiment if we didn't have the DILIsym results. We would just continue very cautiously to explore the clinical development program of lixivaptan without performing these head-on, just hit the nail on the head kind of experiments. It just made our clinical development program much more persuasive, aggressive and persuasive.
Dr. Watkins, did you have any comments on that
Yeah, I was
question?
just going to say, I mean, the DILIsym software, which is because of the initiative is constantly being, improved, is not perfect. It doesn't always predict accurately the liver safety liability. However, with almost no exceptions, that would be a false negative. So, like remdesivir, as Brett pointed out, false positives are almost unheard of with the. So, in other words, the concern that you're going to get a readout of liver safety liability when the fact there isn't there is extremely rare. So it's more usually the other. There's the mechanism that's not in DILIsym, or there's a metabolite you haven't measured, so you get a false negative. Thought I'd add that in there.
Yeah, good point. And I think it's sort of a follow-on to that question. This naturally leads you to the place of what cautions would you give with respect to the use of tools like DILIsym to companies? Maybe we can start with you again, Dr. Watkins, this time, and go the other way around. And just what cautions or pieces of advice would you give to companies who might apply this approach, if any?
Well, just what I said. I think DILIsym has been very useful, as you're hearing from Lorenzo and Vlad, and it's very gratifying, the uptake, and that it's being used in decisions. But it's not perfect, and I think people need to put it in the right perspective that this is an important part, a growing importance of the aggregate data in terms of liver safety.
Dr. Pellegrini, any response?
Yes, I think it's the usual it's the usual situation where you would apply garbage in, garbage out, right? So you need to have very good, solid, strong inputs into the simulation, including good PK data, tissue distribution data. Otherwise, there's too much uncertainty on the results otherwise. So in our case, this was not the problem because lixivaptan was a very mature drug. But I would think in case of a very early stage clinical development program, there may be more of an issue. In that case, perhaps the approach would be to conduct a DILI-sim simulation analysis in stages over an extended period of time as more data become available, refine the result. But again, you need good inputs in order to have good results.
Yeah, that's a good point. Dr. Coric, anything to add?
I'd agree with what was said, and in our case, again, as Paul had alluded to, it's not going to be the end all, be all, data point, but in the backdrop of what is occurring, in certain mechanisms of action, it becomes a critical tool to designing your clinical program the right way. Your clinical program is going to trump everything, right? But it's going to allow you to decide, are you bringing forward the right molecule? And so that's where I find it to be, most useful, in our case, is the level of confidence that we had, engineered out issues in previous generations, and then to augment that DILI-sim modulation with the real life, clinical data to demonstrate that it indeed was predicting that these compounds would be safe. And so, again, I think it's been mentioned, but it's the validation, making sure you have validated tools that you're using, and obviously with DILI-sim, that criteria is met.
Excellent. Well, I'll sort of shift gears here and kind of compare and contrast two sides of the coin, if you will, with respect to drug development. And we can start with Dr. Coric for this one. So, safety and efficacy are sometimes viewed quite differently in terms of proactive investment and focus by companies. It just may be in some cases that they're more willing to invest on one side versus the other. My question for you is just, what are your thoughts on this generally in terms of how you go through the strategy, as a drug development company, of determining where to invest into the efficacy side of that equation and where to invest into the safety side, and balancing those two things? Obviously, you have a finite number of dollars to work with in terms of developing a drug.
I think you're right, but I kind of flip it around. Like, there's no doubt, I think good drug developers, safety unequivocally has to come first, right? And I think you're right. What happens is a lot of people maybe overinvest on the efficacy side of that coin, and then late stage they could get hit on something on the safety side, which is not a prudent way to develop drugs. And so, to the extent that you can incorporate computational modeling and other approaches earlier on to predict safety, is really the wiser choice here. And so, to me, it's a no-brainer. It's discharge as much safety risk as you can, right, before you get to the clinic, to make sure you have the right compounds. And obviously, efficacy is key, but they go so hand in hand. Don't overinvest in efficacy at the expense of safety. Make sure that you're incorporating tools like DILI-sim early in your drug development program. And as far as I'm concerned, DILI-sim should be a standard part of just about any drug's advancement in the early stages.
Dr. Watkins, as a consultant, what's your perspective on companies navigating this efficacy versus safety equation question?
Well, I think, liver safety has a special place in the heart of the FDA, as you know, after the troglitazone experience. John Sr. became an individual who full-time looked for trouble in NDA applications for liver safety. There's never been anybody like that. Now, actually, Skip Hayashi has sort of taken over that role at the FDA. But there's no other equivalent person doing neurotox or kidney tox or cardiotox. So there's always been sort of a special focus on liver safety. And so if you start seeing ALT elevations in a clinical trial, it's very easy to slip into the trying to prove a negative, prove you don't have a problem at that stage. So I think, to avoid, because that's one of the issues. People say, well, no, I'm really worried about the idiosyncratic toxicity, that 1 in 5,000. That's what really kills drugs and everything. But it all starts at the hepatocyte with stress, those mechanisms that are in DILI-sim. If you can get as far as all the data suggests, if you can get a dosing regimen and a molecule that doesn't cause stress to the point of ALT elevations, you will not have a problem with the delayed idiosyncratic hepatotoxicity. So I think, liver safety has been an area that continues to be of great focus. I've actually been part of meetings where with a new drug has had a division meeting because there have been a couple of deaths, one from anaphylaxis, and the entire meeting was talking about whether or not one case was a Hy's Law case or not. So blowing past the anaphylactic death to start, is this a Hy's case or not? So I think liver safety has a particular soft spot in the FDA and continues to be the case. And DILI-sim modeling, is helping address that need from a sponsor standpoint and from the FDA standpoint.
Anything to add, Dr. Pellegrini?
Just a very quick comment. I can speak about the experience with our competing drug, our comparator drug, Tolvapden. In that case, very clearly, safety and efficacy go hand in hand, because if a patient cannot have access to the drug because of the REMS program, because of all the rigorous, very frequent liver function testing, then if you have a compliance issue, you have, because of safety, you also have an efficacy issue. Patients who do not get the drug, they don't benefit of the efficacy of the drug. So we can make a very powerful argument there that we're not just dealing with safety differentiation, we're also dealing with efficacy differentiation.
Yeah, that's a very good point. And I think Dr. Watkins made a point many times that oftentimes what we call a lack of efficacy is really a lack of the ability to push the drug high enough in the clinical studies to get to efficacy, because you have a sort of a restricting liver safety, on that particular molecule. So these two things start blending together pretty quickly as we start, working through that dosing selection process in the clinical trials.
It's interesting, Brett, because if you look at, again, going back to Tolvapden, if you look at the dose in the clinical studies that was most frequently used was 120 milligram total daily dose. But then in the real world, people are actually using half of that dose, 60 milligrams. And it's probably due to other reasons, including certain other tolerability issues with Tolvapden. But there may be also a recognition there that people don't want to push the dose too high because they are afraid of liver toxicity. So you have this massive systematic underdosing of ADPKD patients out there with this drug, and we don't know if it works just as effectively as it worked in the clinical studies. It's definitely something that we can work with, on, improve on.
Dr. Watkins.
Yeah, I was just gonna make the point that I think there's a lot of idea out there that all you have to do is avoid certain properties with drugs. There's the Wade-Tongs rule of 3, and Les Bennett's avoid the B, D, D, CS or SC class 2 molecules. But the reality is, as you're particularly going more and more to more difficult to drug target, the very properties that are in those classes that makes them have liver safety liability are necessary to get the drug into where you want it to go. So even with actually the gadgepants, they all had liabilities. They all tested positive to varying degrees in the assays. It's only when you put them all together with exposure and adding the 3 major mechanisms together that you begin to clearly see the differences in liver safety liability. And I thought I'd just make that point.
Yeah, you're right. There's a big difference between an in vitro red flag and a real clinical problem, and that's something that I think DILI-sim is very helpful at. So, well, we really appreciate the 3 of you joining us today. It's been an excellent discussion, and again, I'm sure our audience would echo the same. It's been very insightful hearing from the 3 of you. Thank you again for participating and joining us. And I'm going to hand it back now to Arlene, our host, and she's going to run a quick poll question. So, Arlene.
Yes, thank you, Brett. Does your organization currently use QST modeling in your drug development projects? We'll give you just a few seconds to go ahead and run through that poll and submit your answer. If we did not address your question today, we will reach out with follow-up via email. And again, a big thank you to Dr. Coric, Dr. Pellegrini, Dr. Watkins, and Dr. Hell. We appreciate you sharing with us your opinions and your approaches for QST modeling. And certainly we invite all of our audience to learn more about how our solutions can advance your drug development programs. Once again, the webinar has been recorded and will be available for playback on our website at www.simulations-plus.com. This concludes our webinar for today, and thank you for joining us. Have a great day.